All right, it's 12:01 here, so I'll go ahead and get started. Thanks everybody for joining, and thanks Dr. Bruckerhoff for um, being able to present, although virtually today. We're super excited to have you. So just starting off here, Dr. Bruckerhoff is an assistant professor in the Department of Evolution, Ecology, and Organismal Biology at The Ohio State University. Her research applies concepts from community ecology, population biology, and landscape ecology to inform the conservation of freshwater systems. She specifically is interested in understanding how anthropogenic stressors at multiple spatial and temporal scales interact to drive community structure and function in aquatic systems. Much of Dr. Bruckerhoff's research also aims to understand the role of biotic interactions, especially with non-native species, in driving population and community change across landscape gradients. She uses a wide variety of tools, including comparative field studies and experiments in the field and laboratory and simulation modeling to address questions that inform the conservation of freshwater systems. And with that, I will pass it over to you.
Great. I really appreciate that nice introduction, and I really appreciate the invitation to speak with you guys today. Let me just share my screen here. Okay. I'm going to assume you guys can see a PowerPoint, and I'll just wait till someone yells at me.
We see the notes slide right now, actually.
Oh, darn it. Okay. Let's try again. I moved it over here.
We see both of them together, is what I should say. So.
Worked so well when I practiced. How about that?
Yeah, perfect.
Great. Again, thank you. I really appreciate the opportunity to speak with you guys today. Today, I'm going to be talking about kind of some research that's been kind of coming in and out of my research program over time, thinking about how stream communities respond to drying. I'm going to start with a really obvious statement that seems kind of silly to say, but freshwater fish need water, right? We all hopefully know this. It's pretty obvious, but it's really important and critical because one, there's not that much water, fresh water on Earth. A lot of people are probably familiar with this concept that freshwater is a very small proportion of the water that we have available. And of that, our surface water, and of that, our rivers, it's a really small percentage. So in general, there's just not a lot of riverine habitat out there. So fish need water, but it's also not trivial trying to figure out how much water fish and other aquatic organisms need. So a lot of work has gone into trying to figure out what's kind of the minimum amount of water a fish needs. So this figure here is showing the proportion of index flow, which just essentially means like the average amount of flow in a river that we can remove before we start seeing adverse effects on some hypothetical ecological metrics related to fish or other species. So there's a whole bunch of literature out there trying to figure out how much of stream extraction or changes in water flow related to human use and climate change that different species can handle. But it's not that simple. In addition to just how much water is in a river, the when and how of that water is moving through the system is also important. So we typically describe streams by their flow regime, which are characterized by the magnitude, frequency, duration, timing, and rate of change of different types of flow events. So this would be the predictability and variability in things like really high flows and really low flows. We often call flow regime a master variable because it influences all sorts of things in a river. So everything from the morphology and to the biology. We know that most aquatic organisms are very in sync with the flow regime and tend to have life history adaptations that kind of sync with these different flow events. So in the literature, it's really well established that things like life history strategies of fish have kind of predictable patterns of composition in response to different flow regimes. So things that kind of separate out fish along a fish life history axes, like fecundity or generation time, we can actually make pretty good predictions about the dominance of these species based on flow. Another really well-established relationship is this idea of flow alteration. So if we're taking not only water out of a river, like I mentioned before, but just altering the timing of these different flow events, we tend to get shifts in community structure from dominance by native species to dominance by non-native species. This is really well established out in Western streams, especially like in the American Southwest, where we've seen a lot of turnover from dominance by native suckers and cyprinids to introduced species. So we know that fish need water. We have all these really well established relationships between community structure and different species and the flow regime. But really, if we kind of take a step out, a lot of our rivers actually go dry for some proportion of the time. So this is a study, a pretty recent study where they tried to map out the probability of a stream being intermittent, which just means that it doesn't flow for some portion of the year. And we can see looking at this map that there's a lot of red, meaning that we have a lot of streams that are actually dominated by these kind of drying cycles. The systems I'm going to be talking about today kind of lie on a nice gradient of dominance by these intermittent streams, but also kind of having some permanent water. So intermittent streams are really prevalent and they're widespread across the globe, and even more locally within watersheds, they tend to be numerically dominant. But they're really understudied. And this is largely an artifact of we just don't have much hydrologic information about drying streams. So if we think about here in the US, a lot of the information we get about flow comes from USGS stream gauges. And these gauges tend to be on rivers that have water all year round. This makes sense that, you know, if you're going to put resources into putting a really expensive monitoring station out, you're probably thinking you should put it where most of the water is. But we're missing a whole bunch of streams by focusing on these perennial streams. Even within our collection of USGS stream gauges here in the US, we are actually seeing that intermittency is becoming more prevalent. So this is a recent study where using the streams that are the gauges that we have that are biased towards perennial streams, we are actually seeing an increase in the number of no flow days across a lot of these areas, especially in the Southwest, not surprisingly, but also the timing of those no flow days. So when those first no flow days are occurring within a calendar year, they're actually happening earlier. So intermittent streams are prevalent, they're understudied, and intermittency is likely becoming more prevalent due to both water abstractions as well as, of course, climate change. So I'm really interested in thinking about taking a lot of these concepts and theories and well-established ideas that we have from the flow regime literature and environmental flows and trying to think about how we can apply those to drying systems. So kind of taking this mindset of going from the flowing regime to the drying regime of rivers. And one of the easiest first steps to start thinking about a drying regime is to kind of think about streams on this really basic continuum from how much there's standing water, how much drying there is, and whether or not the stream flows. So we have these three really nice, easy, simple classifications of streams that are always fully connected, so they never cease to have flowing water moving through them. Streams that dry, but they retain isolated pools. So they either have pools that are large enough or connected to groundwater enough that they're able to maintain some water. And then, of course, those that go completely dry. So this kind of just provides a nice template to think about how to link drying patterns to communities. So for my talk today, I'm going to kind of think about community responses to drying at two different spatial scales. So on the left there, this would represent a single stream, so kind of one single reach that goes intermittent, shown by those dotted lines, and kind of following drying across these isolated pools within a single intermittent stream. Then we're going to take a step back and kind of take more of a watershed approach and compare community composition across multiple intermittent streams with different drying dynamics. So the three main objectives I'm going to focus on today are one, characterizing shifts in community structure as the stream dries. Again, that's at that local scale. And two, characterizing what I like to call the life cycle of an intermittent stream. So thinking about over the course of a year as we have drying and re-wetting patterns, what do we have predictable shifts or predictable life cycle in those shifts across multiple intermittent streams with different drying regimes? And then last, we're going to explore some applications to management and talk about a case study focused on fragmentation and the role that that can play in these intermittent stream dynamics. So the first study I'm going to talk about actually was completed a few years ago, and it was a collaborative project when I was still a graduate student. I want to give credit to Dr. Garrett Hopper because he took the lead on a lot of this work, and it kind of was a big collaborative effort of us taking advantage of kind of an opportunistic drought. So Garrett's now a new assistant professor at Louisiana State University and really took the lead on this work. So this first objective, we're going to be focused on a prairie system, a prairie grassland system in Kansas. This work was conducted at Kings Creek, which is a river that's part of the Kanza Prairie LTER and Biological Station. So it's a long-term ecological research site. So we have lots of long-term data on the fish communities in this river and was funded by both the LTER and supported by Kansas State. So the first two questions I'm going to focus on are, do abiotic factors drive changes in taxonomic composition as pools dry? And do changes in composition reflect species thermal and dissolved oxygen tolerances? So what we're really focused on here is if we look at a pool as conditions change in it, do those conditions lead to predictable patterns and change in community composition? So we developed some hypotheses based on a conceptual model from Bolton, which you can see up in the right-hand corner, focused on macroinvertebrate responses to drying. So the idea here is as a stream loses its lateral, longitudinal, and hyphoreic connectivity, that we're going to see a decline in species richness of macroinvertebrates. We expanded on this a bit to think about how fish might respond to stream drying and think about traits of fish. So as streams start to dry and lose connectivity among pools, we first expect that we might start to see loss of species from really high temperatures, right? Because usually stream drying is happening in the summer, and low DO. Where eventually, as the species are lost, we're only going to have our most tolerant species left in these pools, all the way to complete mortality of all of our fish species. So we had a unique opportunity to study this in response to an extreme drought. So the figure you're looking at now is the number of days with flow in Kings Creek across time. So we have everything where every single day of the year we see flow in the river down to very few days where we're actually seeing moving water. One thing you'll notice is that we've seen a downward trend over time. This is due to both climate impacts as well as vegetation compositional changes at Kanza. But in 2018, where we conducted this study, you can see it's one of the lowest days with no flow on record. And if we look at our long-term fish community data, we see that fish definitely responded to this, and we see much lower abundances of fish during this drought year. So what we did was, like I mentioned before, focused on multiple pools within this single stream. So there's a map of our study area, and you can see our 12 different pools that we tracked. Just to give you guys a visual of what these pools look like, this is tracking pool 5 over the course of that summer. And you can see that we started with water, and we had this nice isolated pool, and it eventually dried by the end of the summer. So we had a good variety of pools that did maintain flow or maintain water all summer, but some did dry. So the first figure I'm going to show you is a community analysis, so just a multivariate analysis, a redundancy analysis of community structure across these 12 pools related to ammonia levels and temperature and pool size. And what we can see here on this redundancy analysis, if we look at the x-axis or the lower axes there, we can see that that first RDA axis described 82% of our variation in community structure. If we look at our should get a pointer here. If we look at our environmental variables here, we can see that temperature and pool volume were really highly correlated with that first community axis, so driving this shift in community structure. So essentially what we see, if you go from left to right, it's essentially the shift in community structures as pools shrink in volume and dry and get warmer. So not too surprisingly, we start off with dominance by species like southern red-bellied dace, which are known to be kind of more cool water species, and creek chubs, and see this shift where we have more of a dominance by species like central stone rollers, to eventually where our main dominant species are western mosquito fish. This compositional change isn't just due to changes in relative abundance of these species, but actual loss of these species like we predicted in our conceptual model. So if we look at these, this plot over here, we're looking at richness as a function of pool volume over the course of that summer. So you kind of have to read it backwards, but as pool volume declines, we see these declines in richness, likely driving those patterns of community structure. In addition to tracking these pools over time, we also did some laboratory experiments to look at the tolerance of these different species to temperature and dissolved oxygen levels. So if we start off thinking about temperature, we just focused on 2 species and acclimated them to several different temperatures. So with the idea that as fish are sitting in these pools and the water's slowly warming, they are acclimating. Where their critical thermal maxima, or the point in which they're going to lose equilibrium under these temperatures, might change with that acclimation period. We saw pretty clear relationships that these central stone rollers had higher CTMs than our southern red-bellied dace. Again, that's not too surprising. That's kind of what we would expect. But if you look at that bottom panel, that's the number of days that in each of our pools where we saw temperatures exceeding these CTM. So clearly temperatures were definitely much above where these species can handle, as we saw driving those shifts in community structure. We also looked at dissolved oxygen. So unfortunately, we didn't have enough oxygen meters to put in all of our pools, but we did do some laboratory tests because we did note that oxygen levels, not surprisingly, were very low at point estimates in these pools. And that definitely matched our community composition, where we saw that western mosquito fish had the highest tolerance for low DO levels. So that top panel represents the concentration of dissolved oxygen in which fish start gasping for air at the top of the water surface. And we can see that our western mosquito fish start doing that much earlier. And if you look on that bottom panel, that's the DO concentrations in which they lose equilibrium. You can see that they could handle much lower concentrations of oxygen, likely due to this behavioral adaptation, which matches what we saw in the field. So none of these results are too surprising, but it was nice to kind of have this nice clean picture of this turnover in community structure, really linked to these physiological adaptations. But this just, as most research does, led to more questions. For me, what happens when the streams re-wet, right? So unfortunately, when we conducted this study, we weren't able to go follow up once the rivers re-wet. So that was a question we were kind of left with this research. And then also, we were only looking at one river, right? So are these patterns predictable across multiple intermittent streams and across a gradient of intermittent streams? So the next objective, we're going to try to answer some of these questions. So we're going to be focused on comparing community responses across a gradient of intermittency. And kind of to simplify it, just grouping that gradient of intermittency into whether or not streams completely dried or dried to just isolated pools. This work was conducted by a master's student who recently graduated. He just successfully defended this work here about a month ago, and he's currently a fisheries biologist at the Bureau of Land Management in Fairbanks. This work was conducted when I was still at Oklahoma State University and was funded by USGS and supported by the Oklahoma Cooperative Fish and Wildlife Research Unit. So the first study I talked about, we were really focused on local conditions in those isolated pools, right? And so we know local habitat conditions can control the composition of fish communities or any kind of aquatic communities. But we also know that there's broader scale regional controls. So by taking a step out and taking more of a watershed approach, we're hoping to kind of account for some of these more regional controls like dispersal and movement among different intermittent reaches. So the questions we're going to focus on here are, do fish communities have synchronous responses to drying and re-retting across the gradient of intermittency? So by synchronous responses, I mean, as a fish community changes within any given stream, is the magnitude of that community change and the direction and timing of that change similar across multiple streams? So we're taking a multivariate, multi-species view of this. So we're going to be looking at a lot of like distance matrices to answer this question. The second question is focused more on that recovery period. So when streams re-wet. So do fish communities, we're also going to throw crayfish communities in for this one, recover, and does that recovery depend on whether or not we see isolated pools or complete drying? So to start off with some hypotheses, these figures represent a year. So we're starting in January and thinking about what happens to the hydrology and community across a year. The blue represents, you can kind of think of it as deviations from mean discharge. So anything closer to 1 is going to be kind of like the long-term mean discharge. And if it goes down, that would mean we'd have lower flows. The orange lines represent community dissimilarity. So if we have 0, it would mean that community is the closest it's ever been to either its long-term mean or some starting point. And as it goes up towards 1, it means that community is becoming more different. So we predicted that in connected streams that maintain flow, that we'll see slight declines in discharge from a hydrology standpoint, and that will correspond with slight differences in community structure. As we move to isolated pools, we predicted we'd see more extreme differences in community structure during the drying period. But importantly, we predicted that these communities would recover once those streams re-wet. And then for the sites that were completely dry, we predicted that we'd see the most extreme changes in community structure, but predicted there might be a lag in the amount of time it would take for communities to recover. So to test this hypothesis, we tracked drying regimes and community structure at 10 sites in the Glover River Basin of Southeast Oklahoma. This is very different from the Kansas system I talked about. Instead of being dominated by prairie grasses, it's a very forested system with very flashy hydrologies. So we picked 10 sites that didn't have any downstream barriers because we wanted to make sure all of our sites had the same potential for recolonization. And we're really lucky that we were able actually to find 10 sites because this is really hard to do in today's world. We have lots of road crossings out there. To track water availability, we modified some hobo pendants. So we essentially just modified the light sensor on these hobo loggers so that it tracked whether or not the sensor was submerged or not. And it also allowed us to collect temperature data. We put these sensors in both a riffle and a pool so that we could track whether or not the sites were connected by flow, if the riffle sensor was submerged, or if the pool had completely dried, if the pool sensor was exposed. We also use wildlife cameras and kind of homemade stream staff gauges to track the height of water across our sites. So we characterize the fish community and crayfish communities at all these sites using kind of classic stream methods. We went out 7 times over the course of the year, so about every 2.5 months, so we could fully get that drying and re-wetting cycle to truly characterize the life cycle of these streams. So the next figure I'm going to show you is going to be a little bit messy, and I apologize, but it's essentially a figure that mirrors those hypotheses figures that I put up, but we have some different colors and we have 10 sites we're going to look at. So I'll kind of walk you guys through it. So the dashed lines at the bottom represent our stage height from our little homemade stream gauges. The red lines represent when a site was fully dry, so no water in the pools. And the salmon color represents when our streams had isolated pools. The black lines represent fish community dissimilarity. So in this figure, we're comparing our community structure to our first sample, but you can imagine a similar plot if we compared it to kind of like the long-term average of our community. So essentially, every time you see that black line going up, that's the most dissimilar your community can be from the starting point. And we assume when there's no fish in the stream that it's at that extreme point. So we ran pairwise Mantel tests across our different sites to compare the synchrony, so again, the magnitude and those changes of community structure and the timing of those changes across all of our different sites. And what we found is that we actually did see some synchrony across our sites that dried to fully dried, where there was no isolated pool. So these 3 sites here had significant Mantel tests indicating that they had very similar synchronous responses in community structure. If we look at sites that dried to isolated pools, we actually only saw synchrony between 2 of our sites. And these sites are actually our 2 largest sites in our system. So I forgot to mention that these sites are organized along a gradient of stream size. So these are our 2 largest sites. So in general, we see synchrony at these sites that completely dry, but if they dry to isolated pools, a little less synchrony. This is probably not too surprising, right? Because you can imagine as you have an isolated pool, that those pools can be highly variable in their size and their abiotic conditions, like we kind of explored in objective 1. What this analysis didn't show us, though, is whether or not these communities recovered, right? So if we kind of look at the tail end of these plots, you can see that most of them fall out right there in the middle of community dissimilarity. So it seemed like they were trying to go back towards 0, but we wanted to more explicitly test whether or not communities were different before and after drying. So we looked at several different response variables. We looked at fish richness, total density, community composition, like in that previous analysis, as well as crayfish density, and developed models that included drying regimes. So that stream classification of whether or not it was fully dry or dried to isolated pools, and drying periods, so whether or not it was before or after drying. What we were really interested in with this analysis, it was the interaction between that time period and drying regime. So whether or not our sites that dried to isolated pools had different recovery or were more prone to recovering than our sites that completely dried. In our community composition analysis, we also include area sample because this model was based on counts and we wanted to control for effort. We were able to include crayfish in this analysis, thanks to an undergraduate student who I want to give a shout out to, Vanessa Rendon. As a freshman, she was really great and identified all of our crayfish for us and really took the lead on this part of the project. So we're going to start with looking at some crayfish results. So this is total densities of crayfish across our sampling period. And we saw some pretty interesting results. So at the beginning of the sampling period, we actually did see differences in community structure between sites that were completely dry versus had those would eventually dry to those isolated pools. During the drying period, we didn't assess that time because there wasn't water in the system. But then if we look at the after period, we actually see no difference between crayfish densities after drying between our 2 different sites. So we did see this interactive effect. So it seems that crayfish did not recover in our sites that would completely dry. Of course, there's the caveat that we weren't able to continue following up on this. So it is possible that crayfish densities did recover post that last April sampling event. Also want to mention that we did try to control for effects of like reproduction by focusing on adult crayfish, but that's pretty tricky to do with crayfish. So we could have some reproductive cycles influencing this as well. If we move over and look to fish, though, there are some interesting contrasts between our fish and crayfish results. Fish densities did not seem to be sensitive to either the drying regime or whether or not we were out there before or after drying. So you can see on that left panel that we don't have any significant differences between our fish densities, although we did see a lot more variability in our at our sites with isolated pools. If we look at richness, we see some expected patterns where we saw higher richness at sites that have the isolated pools relative to sites that fully dry. Interestingly, though, we didn't see any effective drying at our sites that fully dried. So we didn't see lower richness after those sites were, completely dry. All of the species that were there before drying seemed to make it back. The higher richness at our sites with isolated pools isn't too surprising. These are typically larger sites, and just having more of those refugia pools likely facilitates higher richness. So now we're going to move in and talk about community structure. So this is a multivariate response where we're looking at responses of all of our different fish species. We used a method called many GLM, which is exactly what it sounds like. It's just many or multiple generalized linear models. So this is a really nice flexible tool where it allows you to use distributions like negative binomial distributions that you can't use in other multivariate analyses, and gives you an idea of the significance of different variables, both at the community level, and then allows you to explore those individual species responses. So the table you're looking at right now are the community level responses to drying. So for this first analysis, we included both our sites that completely dried and dried to isolated pools, and we're looking for that interaction of before and after drying. We didn't see that interaction, but we did see a significant effect of drying regime, meaning just the core communities between these two sites are different, as well as time. To focus in on that time aspect, because that's really what this talk is about, we actually only saw one species that significantly responded to drying. So yellow bullhead were in our streams before the drying event, but then post-drying, they didn't recover in the time period that we were out there and looking. So in general, most of our other species did recover though. So the significant effect at the community level, we think is being largely driven by yellow bullhead. So this analysis focused on both of our different drying regimes, but we were also interested in focusing on just those sites that dried to isolated pools and exploring the change in community structure during that drying period. So we can't look at that for the sites that completely dried because there's no fish, but we can focus in on those sites that retain water. So in this analysis, we're looking at time period, we're including before, during, and after drying. And we did see a significant effect of this drying period at the community level. If we dive in and look at that at a individual species level, we see a lot of consistencies. So first thing to note as we're looking at these plots is that this analysis, we did include areas sampled to try to control for the change in densities. So these estimates of abundance or count of these different species are corrected for that change in habitat area. So in general, we see higher abundance of most species during the drying period. This perhaps isn't too surprising as, water is leaving out of the system and we only have these isolated pools. There's just less places for fish to go, so they're likely going to aggregate in these isolated pools that are left. What's really interesting about this, though, is as soon as that water comes back, we don't really see any differences between abundance of these different species before and after drying, which I think is one of the most interesting parts of this research. We did have two species that kind of didn't fit this trend. The first was creek chub. You can see there's really big error bars on these estimates. They just were really rare in our data set. And what we think is happening with them, since they're a larger bodied fish, we think they just kind of poke into these intermittent streams, but then swim back downstream. And they didn't seem to get stuck in our pools. So this is an interesting species from thinking about kind of the role that dispersal capabilities plays in these systems, because they might actually be getting out of these systems before they dry. Our other species we think is largely an artifact of sampling. If you've ever sampled benthic darters with electrofishers, they're really easy to miss. They tend to get stuck in the rocks. And we did do some analysis on capture probabilities of all these species, and we had really bad capture probabilities for our orange-bellied darters. That species, we take any results a little cautiously. So just to refresh or review where we are right now, we've characterized shifts in stream community structure as a function of stream drying. We know that communities do shift pretty dramatically, and that's related to their physiological tolerances. And we also know that these shifts in community structure can be fairly synchronous, at least within the most extreme drying regime. So if your stream completely dries, you're likely going to see some similar patterns of community change across time. And what I think is most interesting is that these communities do seem to recover. So we didn't really see that recovery with our crayfish densities, but from a fish perspective, we tend to see very similar communities before and after drying. So last, we're going to talk about this from a little bit more of an applied sense and think about management actions and fragmentation and how that might interact with these patterns. So for this work, we're going to go back to Kansas, or back to the grassland. This again is another collaborative study from some of my graduate time and was led by Dr. Keith Gitto. And what we leveraged in this study was that long-term fish community data that I mentioned before, but also a management action or removal of a road crossing in this system. So road crossings, we know, can be barriers to fish, especially a lot of the small species that are in these intermittent systems. That panel A up there is a picture of this road crossing that even looking at it when it's dry, so this is clearly an intermittent stream, it looks pretty terrible. You can see that giant concrete slab. The next picture is one while that stream is flowing. So this has been a hypothesized barrier to fish movement in this system. The next three pictures are pictures after this road crossing was taken out. You can see some of the geomorphic changes that occurred with that major change in the stream. And then the last picture is really important because this is a picture of southern red-bellied dace spawning right below this road crossing. Again, this is an intermittent reach. It is dry for large portions of the year. But when there is water in it, fish seem to be using it. So that was kind of the impetus for this study, is to understand if fish are able to use upper reaches of this intermittent stream once this barrier came out. So the two questions I'm going to focus on is one, does removing this potential barrier improve dispersal to intermittent reaches, which we hypothesize fish want to get up to? And do densities of fish in streams with and without barriers differ in their response to drought? So that first study that I talked about where we tracked that drought in 2018, that's going to come into play here in this study too. So to look at fish responses to the removal of this road crossing, we used pit tags. So these are little tags that we can insert in a fish, and they are pretty much like microchips you might give your cat or dog. We can then put these passive antennas out in the stream that track the movement when fish pass over that antenna. So this is a complicated mat over here, but I'll kind of walk you guys through it. We tagged all of our fish, so we put these pit tags in down at this triangle point. So this is a perennial reach. It typically always has water in it, except for during really dry times. And you can see the dotted lines that represent the intermittent reaches of this system. The green circles represent where we had these passive antennas. And the red circle represents that road crossing that came out. We're going to ignore this picture of the waterfall until a little bit later for now. But essentially what we did is we tagged fish down here, and then we tracked whether or not they were detected on these two different antennas, both before and after the barrier came out. So these results are not too surprising. This is a figure where the gray area shows when our antennas were out. The upper panel is above our road crossing and then below our barrier. And any color on there represents detections of fish. So not too surprising. Like we thought, just kind of looking at this barrier, it was a barrier to fish dispersal. We didn't have any detections of fish above this road crossing, while we saw quite a few detections of individual fish below this road crossing. The dotted line represents when this structure was taken out, and we see dispersal after this barrier is removed. So maybe this might seem boring and not too exciting, but what I think is really cool about this is how quickly we're actually seeing fish move into these reaches. So the black line represents the discharge in the stream. And you can see that pretty shortly after we see water in the stream after this barrier was taken out, we start seeing detection. So fish are really keying in and using these habitats as soon as they're available. This figure is just kind of splitting this out by species. So that previous figure was just counts of individual unique fish. If we split this out by species, we see we see that this is consistent across the 3 species that we monitored. So that top panel is for all species, and then as we go down, it's for different stone rollers, creek chubs, and southern red-bellied dace. What you can see on this below and our above plot is that we have this big gap here in 2018. That's that big drought year, right? So we all of these reaches were just completely dry this year and that year never had water, so we weren't able to track movement. For our next question, we were really interested in kind of honing in on this drought a little bit more. So now we're going to kind of change to 2 different study sites. So these sites that are circled in green are long-term fish community sampling sites. So these get sampled every single year. We have nice long-term data. And even though this road crossing was removed down here in this intermittent reach, unfortunately, there's also a waterfall, a natural forming waterfall, right below this long-term sampling site. So this kind of provided another opportunity to look at the effects of fragmentation when we're experiencing really severe drought in this system. So if we look over to our these figures over here, the left panel represents this K2A site, which doesn't have any downstream barrier, but of course is intermittent. And then the N4D is the site that's above the waterfall. Before the drought in 2018, we can see that we have fish at all of these sites, and we have this really big drop off in response to that pretty severe drought. After drought, our sites at our fish at the sites that do not have that barrier downstream recovered relatively quickly. So you can see that these most of our species, even a benthic darter species, bounced back pretty quickly. At our sites with this barrier downstream, we do see little blips where we are catching some fish up above that barrier, but at really low densities relative to what they were pre-drought. So this is really important to just kind of show how some of these different stressors can interact across landscapes, right? So we know that fish like to use these intermittent streams. They clearly move back into it very quickly after they re-wet. But if there's barriers, they're not able to access these habitats. So in summary, we see really dramatic shifts in community structure with drying, not too surprising. And these are pretty predictable based on tolerances of different species. Unfortunately, for a lot of these streamfish, we don't have a lot of that data on thermal and oxygen tolerances. But if we did, we'd probably be able to make pretty good predictions about what community structure will look like in intermittent streams. And communities recover very quickly if there's not any barriers. So this theme of trying to both understand drying more across regions and the use of intermittent streams by fish have kind of led me to a couple next steps or future directions that I'm currently pursuing. So I recently received some funding to try to characterize drying regimes across a much larger spatial area. So the idea here is that with different land use, so like the 2 systems we talked about today, one was forested, one was more of a prairie stream with just gallery forests, likely have very just different natural flow regimes, right? And so while we know these landscape gradients influence flow regimes of perennial rivers, there hasn't been a lot of effort into data collection and trying to characterize differences of drying regimes. So we're going to use our cheap methods of using some of these modified hobo sensors to track intermittent streams at a much larger spatial scale. So we're going to be looking at streams across Kansas, Oklahoma, New Mexico, and Texas for some of this work. Another thing we're really interested in is actually trying to apply this to management. So that study with the road crossing is a pretty clear and simple example of just taking out barriers and how they can provide access to these intermittent streams. But there's a lot of other things we can do too. So as part of this broad project of mapping intermittent streams, we're also going to be hosting a big workshop with state and federal managers to try to talk about what we can do to either try to keep water on the landscape or provide habitats in these intermittent reaches. And lastly, something that I'm really interested in and hopefully we'll have the opportunity to continue to pursue is related to this question of the contribution of these streams to populations and communities. So again, they return to these streams very quickly if they have access. We know fish go in these rivers, these intermittent temporary habitats, but we don't really know how important they are for different species population persistence and the persistence of larger meta-community structure. So I'm hoping to get some long-term studies going, both here in Ohio and potentially in the Plains, to try to tackle this really big question of what are the contributions of these intermittent streams to populations. And with that, I would love to take your guys' questions. And again, I really appreciate your time. You can feel free to just yell at me through Zoom if you want, or if you feel more comfortable talking in the chat, that works too.
That was awesome. Thank you for that. I can start us off with a question. I'll give some people some time to form theirs. So I'm curious what your thoughts are on how riparian stream habitat impacts loss of species. Thinking like specifically if you have really altered systems where there's no riparian habitat and maybe more warming, more DO depletion. Yeah, how that might impact the species that you have left.
Yeah, that's a great question. So you already brought up like one of the first things that comes to mind, which is shading, right? And the importance of that. So clearly, if we've had lots of riparian zones, then less shading, and isolated pool is going to get warmer a lot faster and be a lot more productive, a lot more potential for those low DO conditions. Something we brainstormed a lot with some of actually the Forest Service managers down in the Clover River region in Southeast Oklahoma, was the role of forest management in the flashiness of these systems and the loss of beaver in these systems. So this is a big trend in streams all over the place, especially out west, of trying to think about how the role of beavers in geomorphology and keeping water on the landscape. And that's something that some folks are definitely thinking about in this region too. So just those geomorphic changes that come with shifts in riparian habitat are really, really important in all rivers and the resources they can provide. But I think especially in these intermittent streams, they can really play a role in how much water is being retained in that system. A good example from the Kansas system is that they've had a lot of issues with woody encroachment in those prairie systems. And there's been some recent work that has come out that's shown that those woody plants are actually sucking a lot of water out of the streams. So if you already have an intermittent stream and then you have this change in your riparian vegetation, they might become even more dry or dry up for longer. So yeah, great question.
I'll go for another one here if no one else is gonna pop in. So I know like in the US, we've kind of stopped, to my understanding, stopped doing a lot of putting barriers in streams, and it's especially like big kind of dams. I don't know specifically about road crossings, but I'm curious if you are looking to, or your partners are looking to do more barrier removals, or if you're just kind of hitching on to people that are doing barrier removals and looking to study the impacts that have happened.
Yeah, that's a great question. So in the Glover River system in Southeast Oklahoma, we pretty much picked every tributary that did not have a road crossing on it. Pretty much every other one did. About half of that study area is on either private land or co-managed land between timber industry and the state. And then half of it is on a Forest Service land. On the Forest Service side, they have been really active at removing barriers. And there was actually one that's slated to be removed here within the next few years. Unfortunately, with my move away from Oklahoma, I don't, it's a little bit more logistically hard for me to track that, but that is something I'm definitely very interested in. And something that, you know, of course we could do in this region as well. I'd say in federal lands, they're doing a really good job of trying to remove barriers, but it's still a big problem at the county and municipal levels. Amy. I think you're muted.
Yeah, thank you. I was trying to unmute myself and too slow. Thanks. Your talk was really interesting. I was just thinking about people's feelings about intermittent streams. So I haven't worked a lot in intermittent streams, and I'm wondering, I think it's really heartening that like when they dry up, of course, the fish come back. That's really great and a good argument to remove these barriers. Do people tend to think of them as not super important? Do you have to convince people that intermittent streams are important? And how do you do that? And how do managers generally feel about that?
That's a really great question. I'd say like I don't have a good sense from a public perspective, I guess a little bit, like it just in my work in streams in general. If it's a small stream, like landowners tend to say there's no fish in there. That's like one of the most common things. I'm pretty sure anybody who works on stream fish has heard. So I definitely would say from a PR standpoint, we have a lot of work to do, like we do with many conservation issues. But from a like scientific perspective and from a management perspective, I think it's been gaining a lot of steam in the last decade. I mean, the literature is just blown up with studies about intermittent streams, so it's definitely getting attention of a lot of scientists. In the Great Plains region, I was actually pretty surprised when we proposed some of this work about brainstorming management of intermittent streams and trying to track it. We actually got a lot of letter support and people who are really interested in it. So I think there is interest and it's gaining interest. It's just been slow.
Great, thanks.
Oh, it looks like we might have one in the chat.
Yeah, I can read it out loud so we have it on the recording here. Do any of your crayfish species burrow or show adaptations for drought? I worked in an intermittent stream system and a dragonfly species and crayfish that burrowed and used those during droughts. The dragonfly did better in intermittent streams.
Yeah, that's a fantastic question. So unfortunately, the crayfish side of this was a little opportunistic. We didn't design our study to look at crayfish. If we had, we would have used better methods for sampling crayfish. They were just kind of tag-alongs in our fish sampling, but that is definitely important, right? So we would definitely expect, especially in that region, we have a ton of crayfish species that do burrow. There has been some work out of University of Arkansas comparing different species of crayfish and their tolerance to drought. And we've actually seen some non-native species that are more tolerant to drought kind of taking over in some intermittent streams down there. So that is definitely important. Kind of a fun fact just from the fish side, we've had a lot of conversations about the importance of the hyphorheic zone. So that's any kind of water that's below the surface in a stream for fish. We've had experiences where those pit antennas that I was talking about have been buried out in rivers. And we've had students going out and trying to dig them up. They find like darters and stuff down there with the pit antennas. And I know I've heard some other folks talk about doing hyphorheic studies on, like more like chemical studies and like pumping out mad toms and stuff. So there's a lot of work to be done in the burrowing world and hyphorheic zone for intermittent streams for sure.
Okay, can I jump in again? That because I was thinking of that as like where when do the fish go, right? Like they have to go at some point, but like you said, maybe they hang around really until the last minute, and so that's very interesting that they burrow. And yeah, it's not really a question. It's just like that's surprising, and I wondered when they go, and how, I don't know how you'd measure that, but they have to
Yeah,
decide at some point.
that's a really good question. So I actually didn't mention that with my future directions, but we did get some funding. Unfortunately, it's really expensive to like put these pit antennas and stuff. But we're gonna try to replicate that design of looking at fish moving into intermittent streams, but replicate it across a couple systems. So that's actually something we're gonna be starting this summer to answer that question of when do species leave? Do we see differences among species? The burrowing thing, I would love to study that more, but that's more challenging to get at.
Yeah, cool. Thanks.
Is that a pretty like widely known thing? I wasn't aware that fish were doing that or able to do that. That they, I don't know.
I don't know how. I mean, I don't know if I'd say we know much about it. I mean, this is very observational and anecdotal, like how long fish can persist, what kind of conditions they need. I would say it's a very unexplored topic for sure.
Cool.
And like I said, we debate about it of how important it is. Yeah.
I have another one in the chat here. Are the fish going down on their own or being eaten by crayfish in a burrow underground?
That's a good question. I'm not sure. We have found that example of, like I said, we were just like shoveling in streams and seeing darters. Those were live fish, so I'd imagine that they kind of went down there on their own. I will say I am talking about this burrowing, but there has been some experiments with darter species and trying to look at how they do in intermittent streams in artificial streams, like in mesocosm studies, where they have not seen them do that behavior where they were expecting it. So definitely needs to be explored more.
Cool. I have another question as well. I'm curious, like if you have any observations about what happens to freshwater mussels as drying occurs. Like, I imagine that they would not be doing so hot as well, especially if there's a lot of drying. And then curious, like how recolonizing works. Yeah.
That's a great question. So a lot of these reach streams that we're in are very, very small and probably smaller, like below the threshold of when we typically start to see mussels. But some of these actually, like the main stem rivers in this region, actually do go dry too. So that Southeast Oklahoma stream, the watershed right above it is the Kiamiti River. And sections of that have been known to go dry. And that's a river with a ton of mussel diversity, a lot of endemic mussels, and it's been a big issue. That river, though, does have some tributary dams. And so the drying on that river, they've actually linked to, potentially linked to flow releases. So there's some anthropogenic effects there. But that is definitely a big issue, right? Because mussels can only tolerate so much desiccation.
Very interesting. Cool. If there is not any last minute questions here, I think we can end it for the day. Thank you again, Dr. Bruckerhoff. It was an awesome presentation. And happy Friday, everyone.
Yeah, it was really nice speaking with you all, and I appreciate your questions. And again, thank you for having me.